Hi, everybody. It's great to be here. I've been wanting to have a chance to come down and visit Kingston for a long time now, because one of the reasons I'm in Canada is sitting in the fourth row here, Professor Greg Van Anders, who's a great person. So today I will start actually with my own territorial acknowledgement. In our case, I'd like to acknowledge that Concordia University, where most of the research I'm going to be talking about was performed, is located on unceded indigenous lands. The Kanien'kehá:ka Nation is recognized as the custodians of the lands and waters on which the university stands, and Tiohtià:ke or Montreal is historically known as a gathering place for many First Nations. Today it's home to a diverse population of indigenous and other peoples. We respect the continued connections with the past, present, and future in our ongoing relationships with indigenous and other peoples within the Montreal community. I'd also like to just briefly say that I know that territorial acknowledgements can be a little bit of a hot button issue, right? Because on the one hand, I think it's important to think about. On the other hand, sometimes they can come across as a little bit box checky. So I really try to have this in here just to remind myself to keep thinking about this and as a marker that, you know, we're not there yet. This is something we have to keep thinking about as we move forward as scientists living in this complicated society. And the theme of complexities and nonlinearities is going to be very key to this talk that I'm going to give today, because I'm actually going to start by giving you a little bit of my own history. So I'll tell you who I am, say a little bit about my lab, and how I got to Concordia in the first place, because to maybe demystify some of the process. Then I'll give you sort of a broad overview of the kinds of things I do and the kinds of tools I use to do them. And I will give you just a little bit of more detail on a specific research project that we're working on in my lab. Possibly, if I have time, a brief overview of some of the other things we do in my lab. And I will hopefully end with some advice for scientists, especially scientists who are at the beginning of their careers, like grad students. So who am I? This is the best way I can distill myself into a tiny PowerPoint slide full of bubbles. These are the things that I think of when I think of me. You know, I think of myself as a scientist, an author, a friend. I'm queer, and I have something going on in my brain. I was going to say neurospicy of some variety, definitely anxious, but there wasn't room. So something going on there. Almost certainly PTSD, maybe some other things too. We're not sure. But hi, this is me. There's a couple different ways to spell my name. The accent over the E is not French. I can explain the long history behind that sometime if people are interested. It was there before I realized I was going to move to Montreal, is the short version. So my lab is full of a lot of very fantastic people who do most of the research these days while I learn French, write grants, and mentor people. And we think very hard about the fact that we are people who exist within, you know, a complicated universe. And so I have this lab poster that I love. It's modified from a Creative Commons lab poster on the internet, found here. And the purpose is to show that racial justice and support for marginalized communities can't really be separated from the practice of science. And here's where we get into sort of nonlinearities again, because everything ends up being connected, unfortunately, for us as physicists, since nonlinear things are much more difficult to handle, and interactions really are never actually two-body, which is unfortunate. And how do we deal with that in life as much as in science? But it is something that I think is really important to think about. So here I am hammering it again. It's very key to our identity as a lab, I think. So these are the lovely people who do most of the research. And you'll see some of their faces again when I start talking about some of the specific research. But like I said, I'd like to start with a sort of a how do I get here. So I think a lot of times people present the arrival at a professorship as like a really straight line. And as you can see, it was not a really straight line for me. It was a very complicated, nonlinear mess over here. I started, my dad was a professor. He recently retired in the middle of the United States, and that's where I did high school. And then I thought, you know what? I don't need to stay here for college. I'm going to go all the way over to Pennsylvania on the coast of the United States to do my undergraduate, where I studied physics. I studied lots and lots and lots of physics. It was very cool. I got to do just a little bit of research on computer science and physics, so you're going to see some computer science-y things coming in here as well. And then I was like, I love physics so much that I am going to go and do a PhD in robotics. That makes sense, right? Okay, so I started a PhD in robotics at Carnegie Mellon University, and I was there for about 1 semester before the bottom fell out of my mental health, and I was like, this is terrible, I can't stay here. I was absolutely miserable. There was this very, I don't know, the best way I've heard to describe it is sort of an old boys culture almost. There were not a lot of people of color, there were not a lot of gender minorities, they're just, and it was not very supportive. I didn't have a lot of support and I didn't really know what to do. And I was freaking out, and I also just really missed physics, I guess because, you know, I'm a physicist. But so you don't have to stay at the first place you start a PhD, turns out, fortunately for me. But I was fortunate enough that I had gotten into a PhD program at the University of Illinois the year before. And I was like, oh no, I should have gone there, obviously, because everything would have been better. Actually, it was, but I didn't really know what I was doing. But I was very lucky because I called the head of graduate studies from a parking garage in Pittsburgh, sobbing my eyes out, being like, I'm miserable, I don't know what to do. And this man, Lance Cooper, fantastic person, right? I'm not even a student, and he takes 10 minutes to be like, hush, hush, calm down, everything's gonna be fine, just reapply and come here instead. And I was like, okay. So I did, and I got to the University of Illinois, where I promptly started doing experimental research in condensed matter. And looking at supercritical transitions in niobium islands. Now, you may have noticed that I'm not going to be talking to you about condensed matter or experiments or anything to do with niobium. And I did not actually stay in Nadia's lab for very long either. Nadia Mason, fantastic woman, but it turns out I hate getting up at 2 a.m. to play around with giant expensive machinery because I freak out when it breaks and there's nobody that I can call. So I left her lab too. She was very nice about it, and she stayed on my committee. She was my external member. It was great. She's been very supportive. And I went and joined Andy Ferguson's lab. And then I finally actually stayed there for a while and was getting my PhD and was like, I have no idea what I want to do next. Because, you know, I had some people telling me, you should totally go into academia. And I'm like, I don't know. I'm not sure I could really do independent research. I don't think my research is interesting enough. I don't think I have interesting enough ideas to go into academia. And then I was very fortunate because I happened to go to a thermodynamics conference in Edinburgh where I met 2 very important people. One of them was this adorable dog who sat there in the street in Edinburgh and let me pet him while I was kind of freaking out because I was in Edinburgh and it was a lot. The other person was my academic grandfather, Thanos Panjatopoulos, who's a professor at Princeton. And he saw my talk. He'd never met me before. Obviously, he knew my PI, but he didn't know me at all. And he thought that my research was really cool. And he said that my research was really cool. And he also told me, this is awesome. You do great research. You should totally go do a postdoc. And I was like, well, if this person who doesn't really have a stake in me and my success thinks that my research is cool, maybe I can actually do some more research. So I did. I went off to Los Alamos, where I did a lot of research in, well, I assumed it was going to be on, you know, proteins in some sense and machine learning for antibiotic design, and then it sort of was and sort of wasn't. Because at this point, right, I was working on I was doing some molecular dynamics simulations when everything shut down. I had actually just signed my offer letter from Concordia. Everything went to pieces. And my PI calls me and is like, hey, can you model the spike protein? You have a fellowship. You have the time to do this. And I was like, sure, I guess I'm modeling the spike protein now. And I said, how long do you want me to run these simulations for? And he said, just keep running until someone tells you to stop. So I did. In fact, I ran for so long that as far as I understand it, I ended up getting a new policy implemented on how many things you could have waiting in the queue at the time, but there wasn't anything there when I was doing it. And this was good because it was really important research, right? The secret is you can get away with using any amount of resource as long as it is sufficiently important and sufficiently key. The cancer people were a little mad at us, but we were like, look, the cancer is really bad, we agree, but also you have a longer period of time to solve it in. So I think we get priority. And the lab did listen to us on that. We got out some important, good papers. But I was doing all of this research, and I was supposed to be moving to Montreal. So I did nothing to do with moving to Montreal. Unfortunately, my spouse worked out absolutely everything for how to get us from all the way over here to all the way up here with as few stops as possible, because this was pre-vaccine in the middle of the United States, and most of that is the southern United States, where they weren't exactly taking things as seriously as maybe you would hope that they would. So we drove in 3 days from Los Alamos, New Mexico to Montreal. We lost my husband's ADHD meds in Texas. Fun time was had all around, but we made it here. Frankly, I think that there's a reason that there's a Tolkien picture here, and the reason is that it turns out it's really important to have coping mechanisms during very stressful periods of time, and at this point, mine was to dive headfirst into the Silmarillion and tell myself, hey, look, I know I have to go, we have to drive across this pandemic-ridden landscape in 3 days with no sleep, but at least I'm not trekking across an icy wasteland for 30 years. Small bonuses. I also really like quotes, so I constantly repeated the Lord of the Rings quote at myself. All we have to decide is what to do with the time that is given to us. And I think that the pandemic was really eye-opening for me in a couple of ways. So one of these ways was that my advice to everyone is don't start a faculty position in the middle of a global pandemic, which I hope is never going to be relevant again, but that was the first thing that I got out of this. The second thing I got out of this was really I really reoriented my priorities. Like, I do care about research, but I also am realizing, look, I have to take care of myself and I have to take care of my students for us to be able to do any research, and frankly, it's the people who matter, This was just something that was very, very important to me, and I think that this talk, this kind of hybrid talk where I go back and forth, and I'm about to get into the research, I promise, but the back and forth nature of this talk does sort of arise from some of the ways that I was reprioritizing myself during the pandemic. And now I'm going to give sort of an acknowledgement slide, a very unusual one, but just to give people a view, right? Normally when we do an acknowledgement slide, we have like our funding and maybe our lab group and a couple collaborators. This is the acknowledgement slide of many of the people who have supported me, primarily in academe just, so this is missing lots and lots of other folks who have also supported me. But you can start to see, like, over the course of my career, I have had a lot of mentors. I have had a lot of people who have put out a hand to help me. And that has been really important to me. And so I think I always try really hard to help mentor other people in turn, and I also definitely encourage, especially grad students, try to get the support network that you need, build that up. And also, it's not just me doing science, right? Like, when I show you these science results, there's really secretly this whole cloud of people that you never see in the background who are helping me with ideas and with just dealing with the bureaucracy of grad school, faculty appointment, et cetera, et cetera. So this is my first non-traditional acknowledgements. I have a more traditional one at the end of this talk. So that's how I got to Concordia. It was very confusing and a giant mess, but I was able to do that, and I think with most of my scientific curiosity intact, which is great. And so what I'm going to talk to you about now is, where did my scientific curiosity start and where did it end up? So when I was 13 years old, with apologies to my husband who's heard this same talk since I was giving this defense in 2018 in my PhD defense, but when I was 13 years old, I read The Lost World by Michael Crichton, which is a story, much like Jurassic Park, about big stompy dinosaurs. And I thought it was terrifying. And there was a particular subject in this novel that I found particularly terrifying. And now I have a question for all of you. And the question for all of you I have is, given that the hydrogen atom is about 1 angstrom in diameter, just for scale, how big do you think the thing in The Lost World was that I found the scariest? Okay, and I've given you some pretty broad range of options here. How many people, raise your hand, how many people think that the thing that I found the scariest was about 10 angstroms in size? Okay, we've got some takers. Yeah, I can tell that there's definitely some condensed matter people in here. How many people think 100 angstroms? All right, 100, okay. 10,000 angstroms? Yep, and any takers for 10 to the 11th angstroms? We've got a couple of takers, good, I'm glad. I would get sad if nobody takes 10 to the 11th angstroms, because, like, it's cool, even though it's not the answer. So now I picked these length scales for a reason, and the reason is because there are actually a lot of interesting things scientifically in the lost world. Now, the thing that I found the scariest, if you said 100 or 10,000 angstroms, you're correct, because the answer is proteins. But in the lost world, there are various different subjects that come up that are related to science. One of them is small molecule drugs like morphine, on the order of about 10 angstroms, which are given to one of the scientists when he breaks his leg. And then we have the fact that the dinosaurs, which of course are our 10 to the 11th angstrom friends, maybe friends, hopefully friends, preferably friends, they're all, in the lost world, they're all dying of a neurodegenerative encephalitis caused by misfolding of the prion protein. And when the prion protein misfolds, so many proteins fold into specific shapes to function, to perform functions in cells like little molecular motors. But when this thing misfolds, it causes large-scale aggregation into, well, we're not really certain which the damaging part is. I don't actually do research into prion proteins specifically, so this might be slightly out of date. I apologize if it is. But we do see assembly into these fibrils that are on the order of 10,000 angstroms. And this is presumably related, I mean, it's a book, but it still happens, to the neurodegenerative encephalitis that killed these dinosaurs. And when I was a kid, I thought, that is so cool. How does something that small kill something that big? And then I was also like, well, I'm closer to length scale of the T-Rex than I am to the prion proteins. That's terrifying. I don't want to be killed by a misfolded protein. So naturally, when I went into grad school, I started looking at multi-scale molecular mechanisms on different scales, and thinking, hey, how can we, in this talk at least, we look at more than just this, but how can we use small things to kill slightly larger things? We do not look at T-Rex in my lab, which is really sad. Someday maybe I'll figure out a way to collaborate with a paleontologist so that I can say that I've done something on every one of these scales. It did get me a student. I did have one student who was like, I love your T-Rex. I'm sending you a picture of T-Rex. Can I do a thesis with you? And I was like, sure. But so in my lab, we do some work on design of small molecules. Primarily right now we're looking at designing different kinds of antibiotics to kill bacteria, which are a problem, and I'll say more about that in a little while. And we also spend a lot of time thinking about proteins. And often, in our case, we look at the smaller proteins or peptides, many of which can aggregate, not into fibrils generally, although maybe. Okay, strictly speaking, the ones we're looking at do assemble into sort of similar aggregates. They're not fibrils. But these things can actually assemble on bacterial cell walls and puncture those open. And so we actually really are studying things on these various different length scales, modulo the 10 to the 11th one. And we're bringing in, basically we do two big areas of, there are basically two big techniques that we use in my lab. One of them is molecular dynamics, which I have seen many people call an in silico microscope, where typically what happens is you want to understand the shape of a protein or how the protein behaves in different environments, or maybe how the protein aggregates, or what happens if you change various things about the protein. And you do this by saying, okay, we're physicists, so this protein is a bunch of atoms. Those atoms are basically spheres, and those spheres are just sticky balls, and that's fine. And we come up with some set of interactions that describe how these things interact. It's usually just, well, some of them have charges, and some of them we're going to force to be bonded, and those are going to be modeled as springs, because springs are great. Everybody loves springs. And there's going to be some angles as well. And then we're just going to say, okay, well, this is how all of these interact. We have a whole bunch of equations, and we can just solve Newton's laws of motion at every time step based on the interactions of all of these atoms in the system. Now, obviously, this means we're solving lots and lots and lots of differential equations, so this can get pretty computationally heavy, although we've been doing better and better at this in recent years. But we do look beyond modeling things at an atomistic level, where we model every single atom as a little ball. We also look at coarse graining simulations where instead of modeling each atom as a little ball, we model perhaps chunks of atoms as spheres, and so on and so forth. So that lets us get at all of these different length scales by sort of understanding the details of what's going on at a physical level using a computer to solve lots of differential equations. The other thing that we're really interested in my lab is machine learning, because machines are really smart. So you may have seen examples where deep neural networks were shown examples of selfies, and they were able to learn to produce images that look like selfies that might even trick you into thinking they're real. Now, at this point, I really need to start getting into a long ethical diatribe, but I don't have time in this talk. But if you want to hear me talk about the ethics of AI and how it should be used and how it actually is used, I'm happy to do that at some point. But in any case, it's true. Machines are smart, and they are very powerful. They can do really interesting things. They can generate essentially new data points that you haven't seen that you can investigate in detail. However, it turns out the machines are also not very smart at all. For example, this is the panda gibbon slide for everybody to whom I've mentioned this to, so just be aware. For example, you can teach a machine learning model to be able to tell the difference between pandas and other animals. And you can have it trained so that it will see this image and it will say that's a panda. I'm fairly confident it's a panda. But however, you can also then figure out a very small perturbation to this image. Very, very small. So you see this perturbation, this image on the right is a perturbed version of the image on the left, because that epsilon is, of course, a small number. So you take this seemingly random series of slight perturbations to this image, and you or I would be like, well, that's still a panda. It's still a black and white bear-like thing. Looks like a panda to me, boss. The machine learning algorithm will be like, whoa, that's totally a gibbon. Like, okay, that's weird. So what's going on here? Well, one of the things that's going on here is that naively, you probably fed your original machine learning model every image in the form of a giant long vector of pixel values, right? And that's a really long vector. And so this thing is only thinking in terms of all of these little specific pixel values. And that is not at all how we're thinking about it, right? We can just describe it as a black and white bear-like thing. It doesn't take us we don't have to look at every single one of the thousands of pixels to make a decision on this. And that's good, because we can't even tell that there's been any disruption to the image. And that's essentially because we're thinking about this as physicists in some sense. So what do I mean? Well, as anyone who's taken a class with me, there's at least one person in this room who has, knows I really like silly cartoons. This one is from XKCD. And it's about physicists. And one of the things that physicists have a tendency to do is say, well, we're trying to predict the behavior of some complicated system, so we'll just model it as something really simple and then throw in some perturbations. Why does your field need a whole journal anyways? Okay, yeah, it's kind of a running joke, right? I mean, the spherical cow in a vacuum, et cetera. But there is actually something really relevant here, and it's the idea of dimensionality reduction. It's the idea of taking this long description of an image or something else. It turns out that if you have too many dimensions in your system, it's really hard to learn anything about your system, and it's not very robust to small perturbations. But if you can apply physicist thinking or it's not necessarily just physicists, of course, but if you apply physicist thinking to this, if you force a machine learning algorithm to come up with a shorter or a smaller description of something by using a technique called dimensionality reduction, you often get better performance, and you often also end up being able to understand what it's doing better, which is really important. So all that is to say, most of what I do can be boiled down to this slide. In my lab, we do physics-based understanding and design of biomolecules. We bring together artificial intelligence techniques with physics and statistics techniques. And what we really like to look at are, on the one hand, trying to design things, think about the whole design space. So we like to think about the design space, but we also want to traverse through a design space. For example, looking for peptides. Usually we're mostly thinking about designing proteins. And so we often use artificial intelligence techniques to try to characterize these design spaces, and then we often use molecular dynamics simulations to say, okay, well, there are some specific data points that we think are interesting, so let's do a deep dive into what's going on in those. And thus far, we have mostly done these separately, and at some point, I swear, we're going to bring them together. I will tell you how we want to bring them together, but we haven't done it yet. So I'm actually going to describe one of the larger projects in my lab that has two prongs. We're on one, we're looking at the specific system on one side, the search space is on the other side. We still haven't brought them together, but we really want to. And so this is us looking at antibiotic-resistant bacteria being a global threat, and we really want to come up with new candidates for things that will kill bacteria. So that we can both search, we want to map out more of the search space and understand the search space so that we can, and also understand at a microscopic level how different points in the search space work such that we can hopefully and eventually develop novel treatments. One way to deal with antibiotic-resistant bacteria is through the use of antimicrobial peptides, which are short peptides that are part of the innate immune system. And there are lots of them, and typically they preferentially partition onto bacterial cells because the bacterial cells are negatively charged and non-bacterial cells are typically neutral. And then they kill the bacteria, usually by disrupting the cell membrane in some fashion. And it turns out that the buildup of resistance to these things tends to be a little bit slower. Obviously, evolution still happens, so there is still buildup of resistance, but it tends to be a little bit slower because it's not easy to evolve away your entire skin, basically, as it turns out. So this is something we're really interested in, and I really like proteins. So, okay, let's look at proteins that are potentially useful. Awesome. Now, unsurprised, as people are probably aware at this point, there's been this huge explosion of deep learning, and it was really started, I think, in my field in about 2018 with this paper from Alanis Buraguzic's group at Toronto, where they were able to use deep learning to generate new small molecules. And the idea was basically, okay, we have this description that's a string. It's a discrete entity that talks about the molecules and how they're connected. And that's not very good if you want to use the long history of optimization techniques from computer science, because most optimization techniques in computer science like to have continuous search spaces. It's really hard. You can do genetic algorithms, but most optimization techniques that are old or are really powerful like to have a continuous search space. And so they applied what's called a variational autoencoder to create a continuous search space out of these small molecules, and they were able to show that you can optimize in this space. And so, of course, the more facetious way of saying that is, look, we can use a deep learning model to generate new images and trick a person into thinking that a fake image is real. Can we use one to generate drugs and trick a bacterium into thinking that a fake drug is real? Now, obviously, it's not fake. It's a real drug. It's just that can you dream up something new with a machine learning model? And so, again, this comes back to what I've been saying about search spaces and data points. Because you have your search space on the one hand, and then you have your specific data points that you're interested in assessing on the other hand. And that is the big picture project that we're trying to do. So we start, and I'll talk to you about these, we've started by doing multi-scale simulations of things that we already know are potentially good starting points for antimicrobial peptides, because we have experimental collaborators. And we're trying to understand how these things work at a molecular level so that we can determine what the best thing to optimize is, right? Because if you want to optimize, you need to have an objective function. And, well, it kind of kills the bacteria good is not sufficiently precise for a machine learning algorithm. So we're still kind of working on what the best objective function is. So we haven't gotten to these 3 and 4 here. But at the same time, we're also looking into the properties of some of these deep learning algorithms applied to peptide sequences as opposed to small molecule sequences, in particular antimicrobial ones. And then shortly, what we would like to do eventually, I hear that some people do Gaussian process regression. I love Gaussian process regression. Eventually, we want to do Gaussian process regression. The idea is basically once we've determined from these detailed simulations the best objective function, we want to fit that objective function over the surface that we've gotten in step 2 and use Gaussian process regression to travel through this search space in a manner that's where you're picking the best the best things to simulate, and by best I mean, on the one hand, you want to explore new areas because you want to find things you don't know much about, which is good. Gaussian process regression gives you uncertainties, so it tells you where to look to find out more information. On the other hand, we want to look at data points that you can exploit. In other words, data points that are expected by this model to have good properties already, so you kind of do this exploration, exploitation, traversal of the space. As I said, unfortunately, we haven't actually gotten to the GPR yet because we're still struggling on trying to determine what the best objective function is. But I'm going to talk to you a little bit about the work that we've done on points 1 and 2. So we've primarily looked at GL13K and some of its variants, though I'll mostly talk about this today. This is a synthetic beta sheet forming antimicrobial peptide. It's 13 amino acids long, and it's like a small slice derived from a salivary protein. It turns out that its conformations change experimentally in the presence of different membranes and under different pHs, probably because you have these charged lysines in the sequence, and when those lysines will change, the charge on them will change as the pH changes, or when they're screened near the bacterial membrane. And so these things will have been shown to aggregate on bacterial cell membranes, and different variants actually aggregate at different speeds. And so we've done some molecular dynamic simulations. One of the nice things about being in this field is that you can throw up these cute little wiggling protein pictures, which are just really nice. I love them. So this is work done partly by Lindsay Wright and partly by Reza Niknam Hamidabad in my lab. They have done simulations of, first they looked just at these individual GL13K, so they looked at it with charged lysines, and then they looked at it with uncharged lysines for a very broad, very hacky almost version of what happens when you change the pH. They're either charged or they're uncharged. And if you just have one of these by itself in solution, not much happens when you change the charge, which is actually kind of interesting, right? You'd think since they behave differently under different pH, you would see different behavior when you change the charge. You don't. You don't see a change in behavior until you have multiple ones of them running around in the box. I can't show this video without this happens to me every single time. I'm like, oh no, I have to get rid of the laser pointer in order to be able to play this video. Come on. I really need to make a GIF of this video. This is an error. Okay, there we go. But you can see that they do aggregate when they're uncharged on time scales of about 500 nanoseconds. Reza is still working on this. This is also work done by Natalia Watson in my lab. And they showed that really the only change between charged and uncharged is when you have multiple GL13K molecules in solution, and the uncharged ones aggregate, and the charged ones also aggregate a little bit, and this is important. So we were just thinking, well, probably they just repel each other, right, in solution because they're highly charged. They don't. They actually do form transient dimers in solution, and we think this is important because one of the differences in the variance, in this slower variant, has to do with how many charges there are along where the charges lie along the protein. And we did some simulations of them as well, and we're able to show they don't, so the original GL13K tends to be extended when it's charged. But these ones, the ones that aggregate more slowly have kind of this like little hook shape over. And so we think what's actually happening is that in the bog standard GL13K, you have these transient dimers that are forming because they're extended, and they're seeding the aggregation. They're seeding these aggregates so that it happens a little bit more quickly. But this still fundamentally, at this point, as we understand it, comes down to very much just a charge effect. Now, of course, the next question is what is going on with the charges at the bacterial cell membrane? That's an answer for another day because we're still working on it, but it's an interesting question. Now, in the meantime, while my students were performing these molecular dynamic simulations, another one of my students, Sam Renaud, was really interested in looking at variational autoencoders to understand design space of antimicrobial peptides. And so he derived this deep learning, he used variational autoencoders, which are this deep learning model derived from Bayesian probability theory. It's the same basic idea that I mentioned before. You basically take, in this case, a sequence and you forcibly embed it into a continuous space. And in our case, you also hook a property predictor to it to try to force the space, to force the force this model to understand, like to have some knowledge that some of these things are antimicrobial and some of them aren't. And he compared a whole bunch of different models. It was really fantastic work. I'm going to not do it justice because I'm trying not to run over time. But he checked all these different models with all these different dimensionalities. He did dimensionality reduction to boil these things down into 2 dimensions. And what you're seeing here are the original sequences that the model, I think this is training and testing in this particular case. It may only be the testing data, actually. I think it's only, I don't quite remember. I don't want to say for sure. But every one of these blue dots is a sequence embedded into the space, and it's a sequence that was not antimicrobial. The orange ones are antimicrobial, and you can see, so we were surprised, because we actually forced these things to use these spaces to predict whether these things were antimicrobial or not. It cared about those. And so you can see that for some of these models, it pulls them apart, right? Which is what you'd expect. You expect this model to pull apart the things that are and are not antimicrobial if it has to use that information somehow to predict whether they are or aren't. But not all of these models did that. Some of them did really badly on the what we thought was a fairly simple task that we'd set them. So that was interesting. We quantified how well they clustered these things apart. And we were able to show that at least some of them do successfully disentangle amps from non-amps. We also showed, which is a little bit more interesting, we took this model and we said, okay, let's take a point that isn't an embedded amp, right? So we've got points in the space that are not corresponding to things we already know about. Let's decode those and generate new sequences and see, are they likely to be antimicrobial? And we did this in a very, very simplistic manner. Eventually, we are going to go to experimentalists. We haven't done it yet because we're not confident enough that we've refined it well enough to trust it. But we used somebody else's very well-tuned deep learning model to at least check whether generated sequences were or were not antimicrobial. If you take a slice across the most relevant dimension, you can see that thankfully we see that on one side of this dimension, on one side of this search space, the sequences that are generated are unlikely to be antimicrobial. They have about 0 probability of being antimicrobial. And as we move to the other side of the space, you see this partitioning coming into play, and the probability of the sequences that are being generated being antimicrobial rises. We did a lot of work that I don't have time to talk about on seeing whether our dimensionality reduction was actually authentically reducing the space, because that's a big problem. I don't really have time to talk about it, but the gist of it was for our models, the ones that are easier to interpret don't actually do as well on the task that we've set for them. So we have a trade-off there. We did a little bit of trying to interpret the latent space, and we've written out a pipeline that hasn't yet to help us do our next step. But basically, the big takeaways here are that we have these starting points for coarse-grained molecular dynamics that we've been starting to look at. We reproduce the experimental trends, and we can understand those experimental trends primarily through just simple charge effects. And we also see that the latent spaces, the design spaces associated with these different models, tend to be model dependent and may demonstrate trade-offs in different qualities. And in the future, we're going to keep looking, and hopefully we'll get to this Gaussian process regression portion, because I think it'll be really interesting if we can really close the loop on this iterative design model. So in my lab, we are looking, as I said, that's the deep learning search spaces here. We also are doing a little bit of work on RNA targeting antibiotics. This is work that Vrinda is spearheading in her PhD. We are also looking at disulfide-containing proteins and polymers, both toxins, which are just so cool. I love toxins. Toxins also appear in the Lost World, surprise, surprise. But those things bind to receptors. They interfere with neuromuscular transmission. They're really hard to model because they tend to have these covalent bonds that snap them together, and that's very hard to model, as it turns out. And also polymers that also have these bonds that cross-link them together that are interesting for biomaterials design. And we also do a little bit of really off-the-wall research that has arisen out of collaborations. We're doing some, right now we don't have a student on this, she graduated, she did a lot of good work, but we do some work on modeling pigment protein complexes, and we're doing this really fun, trying to get off the ground this really fun research project on using deep learning networks for data augmentation to hopefully better understand brain imaging of long COVID patients. These are in collaboration, which is why they're completely out of my research area in some ways. But we do know a lot about networks, right? And we love networks, and I love networks, so that's why we're doing this. So I'm going to skip that. I just want to say a little bit, I just want to kind of finish up with what I consider to be a few more important considerations for burgeoning scientists. So the first thing I'd like to show you is my incomplete list of times that I have really screwed up. So since 2015, when I started my final research project and found that my code was pulling garbage because I thought, oh, I'll just make it a little bit faster. What could go wrong? Well, what can go wrong is that you're actually not looking at your data anymore. You're just looking at whatever happens to be in junk and memory. Whoops. And going on and on and on, including, oh wait, there was a paper I was supposed to write? What? Wait, I need, so you can see, I mean, these things have changed a little bit, but you can see that I keep having, there keeps being screw-ups. This is an incomplete list, and sometimes it's the same screw-up, right? I got the grant number wrong for the student bursaries in 2021 and 2022. Whoops. And that's actually something I care about a lot, right? I want my students to get paid, but still. And then here's my incomplete list of things that I failed at. So most of these are fellowships or grants. Again, these are incomplete. I can keep adding to them for ad infinitum. What point am I trying to make here? That on the one hand, screw-ups and failures happen to everybody all the time, constantly, and they also suck. A lot. And in science, I think, especially in academia, it's really common because you have to try so many things and you're doing stuff that's so hard that sometimes you just bang your head against a brick wall and get really, really sad because you really wanted to get an anisotropic potential working in HUMD. This is only for Greg. And you can't, after 3 months, and it's just very tragic. And this tends to feed into things like imposter syndrome, maladaptive perfectionism, and catastrophic thinking. I can tell you I have all of these all the time. This more commonly affects people coming from minority communities, but it's up to 70% of folks in STEM, so I think it's very surprisingly common how much this affects people. And imposter syndrome, and it's often associated with perfectionism. It can interact with stereotype threat. You have all of this. It's everywhere. It's really hard to deal with, and we don't talk about it a lot. So I just want to tell everybody, including myself, you're a person with worth outside of your work. Your entire being, if you're like me, you might sometimes think, oh no, my entire being is caught up in this one research project, and if it fails, who even am I? And sometimes with me, I'll get, it'll interact with the imposter syndrome. I made a mistake, and now I'm revealed as an imposter. I made a mistake, now I'm not deserving of whatever. I made a mistake, and the only way I can atone for it is feeling really bad. None of this is true, but my brain tells me it all the time, and so I try to keep combating it with telling myself and telling you that you're a person with worth outside of your work. And listen to Jean-Luc Picard. I have to keep repeating it to myself too. It's possible to commit no mistakes and still lose. This is not a weakness. This is life. I know it's a ideal. It's a saying. They're always very pithy, but it is I think in terms of quotes. So there we are. Coping mechanisms are hard. I have a lot of them. You probably have to figure out your own. Ali Brosh, read Ali Brosh. She's great about this kind of thing, believe me, just totally gets it. I do therapy when I can find the time. I do lots of Dungeons and Dragons. So, if you like D&D, come talk to me. I never have spoons a lot of the time. I do meditation. I know that doesn't work for everybody, but I like to give it a shout out because I was really surprised that it actually did work for me. It helped my brain. So it's not a myth. It can work for some people. It's better to satisfice than optimize. I keep saying this, but a lot of support, just like so much support from so many people. Ask people for help, I'm not kidding. You know, the worst they'll say is no. If they're a jerk, if you ask them for help and they're a jerk about it, that's not on you, that's on them. You just walk away, you're like, okay, you're not a person who's gonna help me. But so many people really will help. Oh yeah, my favorite piece of advice, which is just ignore anything I've said if it isn't useful for you. Ignore advice that isn't right for you. This was advice I got from, I think she was the chair of engineering, mechanical engineering at Urbana-Champaign during a talk. It was really good advice. Sometimes you really can just throw out advice. You don't have to listen to everything that everyone says. They don't understand your situation as well as you do. Sometimes it really helps when you're going to ask people for help, when you're going to ask for advice, telling them the kind of help you need. Like, do you need them to just sign a form? Do you need their advice? Do you just need them to say, man, my brain does that too, that sucks, you know? People don't always know the right way to help, and it can really help them help you. If you don't know, that's fine, but if you do know and you just tell them right out, look, I just need you to tell me that it's okay. I just need you to pat me on the back. I just need you to listen to me cry for 30 minutes about how I screwed up entering the students' grades again or something like that. And I also do a lot of creative writing. And I think that's it. We are at the more traditional acknowledgments of all of my various funding and my computational areas and my absolutely wonderful lab without whom I could get nothing done, including this photo, which was, I think it was Natalia's idea. Isn't that neat? It's such a pretty spread. They did such a great job organizing this whole thing. So thank you very much for listening, and I would be happy to take any questions.
Thank you so much. Yeah, I have to stop the recording.
Oh, yeah. Thank you. Yes, absolutely. Is it stop?